don't know how to add her. She wanted to see if she could study with us. I was trying to tap the thing on my phone to add her until from the contacts, but it didn't work.
Huh? Oh.
Oh, I can put it in.
But for that.
Oh, it's the.
Oh, yeah.
All right, we'll get started. Before the break, we talked about DNA replication. Now we're going to talk about transcription translation, making proteins from the genes. Let's clear the board. So DNA carries the instructions on how to make the proteins or polypeptide chains. The relationship is one gene, one protein. Okay. Genetic information in DNA is conveyed in linear sequences of those four bases of the DNA: the A, the T, the C, the G. Okay, so it's linear. The gene and its polypeptide products are collinear. Okay, you just read it, you base pair in a row. The gene is a collection of adjacent nucleotides that specify the amino acid sequences for one polypeptide chain. So the corresponding nucleotide and amino acid sequences are collinear. Okay, so I say, what's collinear? I always kind of like give the example of this: amino acids from nucleotides to amino acids with, like, say for example, let's say your secret code is the alphabet and it codes for numbers. Okay, just as a simple fun example. So the code is the alphabet, A, B, C, D, E, F, G, all the way to Z. And let's say that this secret code is coding for numbers. And let's say the letter A codes for number 1, B for 2, 3, and 4. So what does Z code for? 26. Okay? So that's it. That's our secret code. And if you wanted to, like, make a secret code using your alphabet, there it is. That's my secret code in these little triplets. So then you decode it. Okay, what's my number that I'm coding for? 1, 1, 2, 3, 4, 3, 26, 1, 2. Okay, I mean, and then I did it linearly. I did it from one way to the other. Okay, right? It's not random. I didn't jump all around. I went in a line, and my red number was collinear with my alphabet code. That's all it means, okay? So when you decode, you don't jump around, you don't go in some weird order, you just go in a linear fashion. However, there's a problem with my code. How do I code for number 27? I can't, because there's only 26 letters in the alphabet. So do you see the limitation? Now, there's a similar problem with nucleotides and proteins. It's like, well, I'm showing you here. How many bases are there in DNA? 4. The A, the T, the C, the G. There's only 4. Now, how many amino acids are there? There's 20. So how do you code for 20 things from 4 molecules? Well, the one thing we could say right off the bat, it's not a one-to-one relationship. Meaning, thymine can't just code for one of these. Because if it was one-to-one, how many amino acids could you code for? 4. But how many are there? 20. So how many are left unaccounted for? 16. So obviously, it's not a one-to-one correspondence. And, you know, they worked it out. And correspondence of, between nucleotides and amino acids, not one-to-one. mRNA is used to make codon assignments, and the genetic code was fully deciphered in 1966. So let's remember our years. 1953 was when Watson and Crick published their first paper to Nature. 13 years later, they figure it out, how it works, right? So here's the genetic code, and what it is it's not one-to-one. Each nucleotide codon is a triplet. Okay, if you do it by triplets, then you have all these permutations. And if you do the math, there's all codons contained in 3 nucleotides, not 1. Amino acids are specified by more than 1 codon. So a codon is 3 mRNA bases. Okay, so for example, notice that the code we use is for mRNA codons. That's the first term to get. They're triplets. There are 3 nucleotides. For example, look at the top left. Uracil, uracil, uracil. Uracil, uracil, cytosine. Okay, UUC. They're different, but they both code for 1 amino acid, phenylalanine, PHE. So these are 3-letter abbreviations for the amino acids. But this is 1 amino acid, and the code to put it in a sequence is either UUU or UUC. Okay. So how you read this table is let's say you have a codon of GGA. What's the first base of GGA? What's first? Well, G is first. I'm just telling you how to read the table. Let's just read the table. How do you find GGA out here? Well, you could just kind of randomly look, but it's organized for you to make it easy to find. So you see how it says first base? What's the first base? G. Oh, this row, so it's here. What's the second base? G. So you go G, G, it's here. What's the third base? A. So here's GGA. What does it code for? Yeah, the GLY, that stands for glycine, I think. But anyways, that's how you read the table. Okay, but you have to be given the mRNA codon, and then you can kind of figure out what it codes for. So these are our bases, and this is our amino acids. So altogether, 61 of these codons specify for amino acids. There's many more than you need, because how many did you need? How many amino acids are there? 20. So you only needed how many? 20. You only needed 20. But how many do you have? You have 61. So you have more than you need, and they call that the degeneracy of the code. That simply means, 2 codons can code for the same thing. 4 codons can code for the same thing. That's all they mean. You got more than you need. That's all it means. However, 1 AUG, that's a start signal to start making the protein. And you have some that signal to stop making the protein. So some are start, some are stop. We're going to move on. So that's it. That's the code. Okay, so DNA codes, but how? Okay, so let's see. Let's make this half sheet question number 2. Answer that question. Where are proteins made? You got a 50-50 shot. What do you think? Where do you actually link the amino acids together? The answer is B, in the cytoplasm. To make proteins, does DNA ever leave the nucleus? Let's make this half sheet number 3. I hear one vote for no, and anyone you think yes? Does DNA ever leave the nucleus? No, it doesn't. The no's have it. DNA never leaves the nucleus, but it has the instructions. So DNA has the code. Proteins are made in the cytoplasm. So, you know, how do you go from DNA to protein? And I've kind of already answered my own question. I just want to see if you can remember for the break. Which kind of RNA is it? It's the M1, right? So you need a molecule to decode the genetic message. That's transcription. Then you need a messenger to kind of like take it, a messenger molecule to take it from nucleus to cytoplasm. So RNA decodes and carries the info from DNA to cytoplasm for protein synthesis. The DNA is located in the nucleus, whereas protein synthesis occurs in the cytoplasm. The genetic information, the nucleotide sequence, is decoded by that mRNA, I just say RNA here, which would then move into the cytoplasm where it orders up amino acids. Okay, so RNA, how did they know it was this? We take it for granted. What they kind of observed before they figured all this out between 1953 and 1966, they say that, oh, well, cytoplasms of cells that make lots of proteins, they always have lots of RNA. Okay, so that's kind of a correlation. And they noticed that DNA and RNA are very similar, right? We learned that today too. All the bases are the same except for what? RNA has the U, okay, but DNA has the thymine, the T. So they're very similar. And you could just kind of base pair that one substitution. And so also they noticed that DNA is basically double-stranded and RNA is simply a single-stranded version of DNA. And so this kind of leads them to think, well, DNA probably has something to do with making the proteins. And so this became the relationship between DNA, RNA, and protein. And they came up with this idea as early as 1953. It's called the central dogma. Of molecular biology. A dogma is like something you passionately know to be true. It's like a principle. It's the core belief of your religion or whatever it is you believe in. Well, it's DNA, RNA, and protein. DNA is the code. Now, to get the code out of the nucleus, you have to transcribe it. The result of that is an mRNA molecule. Yeah, I just say RNA, but then you have to like get from the nucleotides to the to the amino acids. So that's, you're translating the code. So you go from DNA to RNA to protein. That's basically the central dogma here. And this is the picture we looked at earlier, which kind of shows the central dogma. DNA to RNA to protein, right? Where's the protein? Polypeptide down here, right? The DNA is still in there, it never left, but the mRNA acted as the messenger to carry it there. Transcription, translation. Now, I taught 3 different forms of RNA. So let's pretend it's way back when and they hadn't identified what's what yet. What they did know was that there was 3 types of RNA, and they kind of like could quantitate how much RNA was in a given cell. RNA number 1 which was the most plentiful of the RNA, and they said most cells have 80% of this RNA. RNA number 2 is about 15% of cellular RNA. The lowest percentage, about 5%, number 3. Okay. And I asked students, well, what do you think, which one's the mRNA? If you had to guess based on the percentage. What do you think? It's just a guess, there's no wrong answer by guessing. I'll show you what it is. Turns out it's this one, 5%. So a lot of students guess, oh, it's 80% because the function is so important. But just in terms of the amount, it's the least amount. So what they found was, it's on the next slide here. The rRNA is 80%. The tRNA is 15%. Remember, that delivers amino acids to the ribosomes. And basically, the messenger RNA is a very small percent. So RNA, you have a lot of pictures that kind of show you the process. Here's one of them in the book. We'll go through the details. But these are more for you to kind of like read in your book. It's hard to teach from. But you see all the RNAs coming together. You see the mRNA being formed as a result of transcription. And then you see it coming out here with the rRNA. And you see the tRNA transferring the correct amino acid. So they all come together in this process in that figure. So we'll take it step by step. But I'm going to give one slide to the role of enzymes. Because in 1953, when Watson and Crick came out with their paper, the geneticists, their kind of like quest was over. Their 50-year quest was over. They figured out which molecules carried the genes. But then if you're going to figure out how the cell biology works, that's a job for biochemists. They have to like figure out how all the enzymes and how all the chemistry works. So there are enzymes involved in transcription, and they're called RNA polymerases. These are the enzymes that make RNA. Okay. One condition for them to work, to make RNA, DNA must be present. That makes sense. You're not going to make an mRNA if DNA is not there. So it's pretty intuitive. Well, here's a picture from the book. This is transcription, and it's the initiation part. To make an, just think, why am I showing this to you? The goal is to make mRNA, right? Because that will have the code. And it starts with the enzyme, the RNA polymerase. It binds the gene at a region called the promoter region. The RNA polymerase, that's the enzyme, binds promoter region. That's what they call initiation, okay? With the help of the transcription factors, RNA polymerase binds to the promoter and pries apart the DNA into two strands and initiates the mRNA synthesis. mRNA synthesis initiated. Moving on. Elongation. Step number one was initiation. I should probably title it. Step number two, elongation. Let me read what it says on the slide. As the mRNA polymerase moves along the template strand, elongating the mRNA transcript one base at a time, it unwinds the DNA double helix before it rewinds the double helix behind it. So it's unwinding, rewinding. Basically, you're base pairing with the DNA template strand. Base pair with the DNA template strand. So DNA is a double polymer, right? So if you have two strands of DNA, you base pair with one of them. You call one the template strand. Call the other one the coding strand. Coding strand, template strand. Remember, that's DNA, what I drew in orange. But when you base pair, I mean, you're base pairing only with the template strand as you elongate in one direction. Okay, so my blue line, that's my mRNA transcript. So if I'm base pairing with it, I know the sequence of the template strand. Okay? So that's kind of all you're doing there. You base pair with the template strand. The other one's called the coding strand, and then termination is just you're done. You hit the stop signal, and you're released. mRNA synthesis ends when the termination signal is reached. RNA polymerase and the completed mRNA transcript are released. Termination, mRNA is released. You have to clean it up. I mean, there's some processing of the RNA that has to be done, but for the most part, we won't consider that. Okay, so given the DNA coding strand, so I give you some triple letters there. What number on the half sheet are we on? 3 or 4? Are we on 3?
Oh, 4.
We're on 4. Yeah, I think 4 sounds right. Number 4. Number 4, I give you that. ATG, CCC, GAT, TGA. And I'm calling that the coding strand of the DNA. Now on your half sheet, I want you to write 2 things. The template strand and then the mRNA codon. So go ahead and take a minute to do that. All right, let me check back in. I think that was enough time if you're following us here. If that's the coding strand, and I'm asking you to make the template strand, we're just going to base pair with DNA bases. So, ATG, G should have been T, A, C, G, 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 C, T, A. There you go. Okay? This is DNA template. Then I asked you to base pair like transcription, right? Initiation, elongation, termination. Well, basically, now let's make the mRNA. You base pair with which strand? Yeah, the template strand. So let's just base pair with this red strand. I'll use the blue. So, but now it's RNA. So what goes with T? A. What goes with A for RNA? Yeah, the uracil one. Boom. That's G. What is this? C, C, C. What is this? G. U. Yeah, it's U. This is U again. G. It's the last one.
A.
Now what is this? The blue? It's my mRNA codon. This will carry the code to the cytoplasm to make the polypeptide chain. And we do it in triplets because, well, you know, that's codons are triplets, and a triplet codes for one amino acid. So, I want to pause here for a second. Why do they call this the codon strand? I'll tell you why. This is the coding strand. Look at the coding strand and your mRNA codon. Aren't they the same? Except for instead of T, you got U. That's why the coding strand is the coding strand. Because when you actually make your mRNA, you're basically copying the other strand, except the uracil. Okay, let's move on. Well, here's a picture of what we just kind of did, practice there. Oops, sorry. Template strand, see if you're with me here. Is template strand DNA or RNA? DNA, and you transcribe it to the RNA. So you go from purple to the blue, that's the mRNA codon. So you can kind of do it in practice at home. You got 9 triplets here. Okay, then you have to translate. So that's where the tRNA comes into place. The tRNA base pairs as well. So that's still RNA. However, look what's on the other end of it. The correct polypeptide. Okay, so that's translation. So we're going to kind of use this table again. For half sheet number 5, find the correct amino acid for each of those codes there. Half sheet number 5, find the correct amino acid for all of these. 1, 2, 3, 4, 5, okay? AAA, GUU, GCC, all of these. Read the table. You'll have to do this on the test. Lucky for you, I will provide this on the test. So you don't have to memorize the table. Just know how to use the table. Okay, well, I think if you knew how to use the table, you should be done. What'd you get for AAA? Oh, yeah, that's lysine. Okay, so I did the first one. I'll check to see if you did the rest correct. Remember, these are codons for at the mRNA level. Let's practice again. Number 6, right small, I might be running out of room with that little strip of paper. But I'm going to give you the coding strand. And I'm asking you, all for number 6, if that's coding, what's the DNA template strand? Then what's the mRNA codon? Then what are the tRNA anticodons? And ultimately, what will be the amino acid sequence? And so I'll give you a few minutes for that. It might take you a little while. Okay, you have one more minute and I'll check back in. All right, so this exercise was supposed to get you to like accomplish this figure. Then you start with the template strand, and then you just base pair with that codon, and then you go one step further. You just keep base pairing. All right, mRNA to tRNA. So let's go ahead and do it for the sequence that I gave you. This half-sheet question. I gave you the coding strand. The DNA template strand is just base pairing with DNA nucleotides. So T, A, C, G, 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 C, T, A, C, T. So this is our DNA, this is our template strand. So this is our double-stranded DNA. And when you transcribe a gene, you transcribe which you base pair with which strand? The template. Okay, so then you just base pair this with this red one that I drew, and that's going to be our mRNA codon. So when you're making a codon out of mRNA, it's going to be A, U, G. Yeah. And again, to check yourself when you make your mRNA codon, I mean, it should match the coding strand, except instead of a T, you have U. Okay. Now, this is our code. So to translate the code, you need the transfer RNA. I'll use orange. So you're just going to base pair with the coding strand. Okay, so base pair with that, it's going to be U, A, C, G, 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 C, U, A, C, U. So this is our T RNA, and you call it the anticodon, the base pair with the codon. Now, to figure out the correct amino acid sequence, what you got to do, common mistake students do is like, you read from this bottom row, and that's incorrect. Because our code, our genetic code is based on the codon, not the anticodon. So which table do you read at? Which level do you read at? The blue on mine. You read from this one. Because they call it the codon because it's the code. The anticodon is just base pairing. Our amino acid polypeptide chain will have 4. Okay, and so basically, don't read UAC, read the table for AUG, which is just methionine, which is the start signal. MET, and then CCC, right here, proline, PRO. Then read from GAU, aspartate ASP, then UGA, yeah, it's a stop signal. Technically, it is not an amino acid. Okay, so it stopped. So our polypeptide chain is like 3 amino acids long. Thionine, proline aspartate, that's it. Just remember, read the table from the mRNA. You'll be okay. The details of what we just did as an exercise is transcription from this level to this level. But when you go from mRNA to polypeptide, that's translation. And the details of that are on the next slides. You see all 4 components get together. The rRNA, large and small subunits, the mRNA and the tRNA, they all come together. And you have nice pictures in your book that show you this. Right here, you can study these, and let's kind of look at the details of some of these. Here's initiation of translation. Translation initiation. Initiation occurs when all 4 components, the ones I just stated, they actually all come together. All the RNAs come together. The small subunit of the RNA, of the rRNA, the large subunit of the rRNA, the mRNA, and the tRNA. Okay. Now in the picture, do they show this coming together part in the cytoplasm or in the nucleus, where they all come together in the picture? In the cytoplasm or in the nucleus? In the cytoplasm. That's where translation occurs. Where did transcription occur? In the nucleus. That's where the DNA is. Okay, well anyways, here's kind of like details of it. There's the 2 subunits of the rRNA. They have these EPA sites. They're kind of like showing you kind of midway making a polypeptide chain shown here. So the last one they added was proline. The next one coming in to the A site is the anticodon transferring leucine, LEU. So what they show you here my line on the board is their mRNA, okay? They have E, P, and A. E, P, and A. I'm doing 3. Each line is a base, right? The E, the P, the A. So basically, here's my polypeptide chain. Attached to my tRNA. So the tRNA is base pairing. It's base pairing with my mRNA. Okay, so the tRNA has the anticodon. The codon is what I'm reading here. Okay. the chain you're making is at the P site. You want to bring in something new, it comes in the A site. So incoming is for the A site, the P site is what you're making. You use the E site to kind of eject things, so I'll kind of show you step by step. So they're making LEU, that's GAU. It's going to match with CUA. That's the code. I'm going to backtrack. Just remember, CUA. Let's read the table. CUA. CUA is right here. What is the code for? LEU, leucine. I'm going to jump back to where I was. So there's CUA and there's leucine, right? That's the code. It's bringing it in, okay? So it comes in. There you go. You went from there to here. There you go. You're going to add your leucine to the chain, okay? Then what are you going to do? You're going to move. You're going to translocate. So they call this peptide bond formation. I should probably write that as a step. I don't want to try to draw anything else. I just wanted to give you a sketch of what they're illustrating there. The EPA stuff is coming in. But when you form the peptide bond, what are they talking about? What peptide bond? Remember, that's the bond between amino acids, right? So what you have to do, if you stare at this long enough, you probably get it. Here's the last one you added, proline. And what's the new one you're adding? Leucine. So you got to put a peptide bond with that. Right there. See that? New peptide bond? That's what I'm referring to. Okay? So peptide bond formation. Then what do you have? Then you have what's called translocation. You move in this direction. So let me describe what's happening. As the entire ribosome got cut off there, it shifts by one codon along the mRNA. That's essentially what you got to know. So you're going to shift so that CUA now shifts to the P site. It was in the A site. Okay, shift one codon. So basically, just focus on CUA. Our CUA was in our A site. But when you shift, the CUA moves to the P site. So instead of being at the A site, it's at the P site. Okay, that's essentially what they're showing you. You're just moving over one. So now you can add another one if you want, or you can stop the thing. You move along. Now you've moved. Now you have a new one exposed. That's the CUC code. Okay. Yeah. This CUA moves to the P site. That's it. That's it. Okay. And when you move over, well, you're moving to a new one. All right, well, let's see. You keep moving and moving and moving until you get to termination. The determination is when you reach a stop codon. They call it a release factor. So you're not adding anything new to the chain when you get to termination. So termination is when you reach a stop codon. That's what I'm writing here.
Yes,
Yeah.
what is EPA?
The EPA refers to, on the rRNA, the E, the P, the A. They call them EPA sites, okay?
Are those like active sites?
Yeah, they're the sites where the mRNA, the tRNA are able to bind to each other. Okay, so boom, release factor, done. You got your polypeptide chain. And now what? You have a newly formed polypeptide chain. Now you use your Golgi, your ER Golgi system to process it. You've made your protein. I mean, we learned a lot about the process, but the thing you made is the important thing. It's kind of like when I get a new iPhone, I'm really excited. Ooh, I got a new phone. Ooh, do I care how they made it in China? No, but it's really important. We just learned how you made it, okay? But then you get your phone, I can't use it. Don't I have to I hate programming new phones. I hate new phones. It takes so long to get used to. But it's the same thing with proteins. Like, you have to process it before it's ready to be used, just like getting a new phone. There's a lot of things you have to do to it. Right now, it's just a polypeptide chain. And well, let's see. They show you this polyribosome. And what you're looking at there is a bunch of RNAs. Well, let me see if you can figure this out. What are these like little red globs? What kind of RNA? The rRNA, the tRNA, or the mRNA? What are these little red globs there? What do you think those are? The rRNA. Remember I said how many subunits do they have? Two. One's large and one's small. That looks like a small one. That looks like a large one. What do you think the blue line is? mRNA. You just move along there, and these little worm-like things, that's our polypeptide. And once you complete making those, there's what it looks like in a real image. Then you kind of have some processing to do. And what happens? Well, some just enter the cytoplasm. Many enter the endoplasmic reticulum and move through the system. They're modified, they're packaged. They are newly formed, and, well, they contain a single recognition peptide that directs the mRNA ribosome complex towards the ER. Okay, like I said, you got a new phone and you had to do things to it to get it working. So you basically, okay, let's go through the steps here. Let me summarize this for you. Your new polypeptide chain, it has a signal, okay? Where is that? Okay, here it is. It's called the signal recognition peptide. The first thing it does in step one is the signal recognition peptide, it binds to a receptor site. This is the first step. It has to get into this complex. Write that down. I'm just going to say the polypeptide chain must enter the Golgi, I should say in order, the ER/Golgi complex. Earlier this morning, we defined that as the endomembrane system, as comprising the ER and the Golgi. So what we now know is that the new polypeptide chain has a signal recognition peptide or SRP for short, that allows it entrance into the ER. before processing. And that's all I really want you to know. The details of which you could read for yourself. But just how does it get in there, the SRP. And that concludes the material for Chapter 3. So you haven't finished the homework yet. You guys know when the homework's due?
Thursday.